Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Today's topic is polymerase chain reaction. In this you will be learning about what is polymerase chain reaction, the principle of PCR and what are the different components taken for PCR reaction and the steps in PCR reaction, the three steps mainly the denaturation, annealing and extension steps. And in the last you will be learning about the application of PCR. Dear learners, let's study about the PCR reaction in detail. The polymerase chain reaction, or simply PCR, is used to amplify the DNA molecule. The invention of this technique is a breakthrough in the field of science and technology, and you will see that this technique is a simple, rapid and inexpensive tool, and it is used to generate millions of copies of DNA from a small amount of DNA. This technique was first developed by Carey B. Mullis in the year 1983, but this person was awarded Nobel Prize in Chemistry in the year 1993 along with other scientists called as Michael Smith. If you see the principle of the PCR technique, the whole process of the PCR, it involves three events, mainly the denaturation, annealing and elongation. These three steps, they make one cycle. Like this, many cycles are repeated to multiply the DNA into many number of copies. And if you see this picture, at the start of the reaction, you will be having a single double-stranded DNA molecule. And if you see the second cycle, the number has increased, doubled to two molecules. The second cycle 4, third cycle 8. Like this, the DNA gets multiplied exponentially and per each cycle, and the continuous doubling, it is mediated by the enzyme called as Taq DNA polymerase. So at the end of the reaction, you will be having millions of copies of DNA, and in each PCR reaction normally 30 to 45 cycles are run. So let's see about the components of the PCR. The first one is the template. So in order to amplify the target DNA sequence, you require either whole genomic DNA or plasmid DNA, and it can be used as template for carrying out this PCR reaction. Next one is the DNA polymerase. The DNA polymerase main function is to promote DNA chain extension, and the polymerase which is used in PCR is Taq DNA polymerase. It is a thermostable DNA polymerase, and this enzyme has stability even at high temperature. So the polymerases which are used in PCR, they are extracted from thermophilic bacteria like Thermus aquaticus, and also from archaebacteria like this Pyrococcus furiosus, PFU polymerase, and Vent polymerase, it is extracted from Thermococcus litoralis. So these type of polymerases which are used in PCR, they are thermostable and they have the ability to resist even at to be stable at high temperature. Okay, coming to the next component, deoxynucleotide triphosphates. So these are the monomeric units, the nucleotide bases like adenine, thymine, guanine and cytosine, and they are the building blocks of new DNA strand synthesis. And if you add these monomeric units in the presence of the DNA polymerase, the chain extension occurs. So all these components, they are mixed in a suitable medium. So for that you will be using the buffer, and these buffers, they provide optimal pH and optimum concentration of various ions which are required to stabilize the PCR reaction. The next component used is divalent cations. So for the polymerization reaction, you required the particularly the magnesium ions. So they will be acting as cofactors for this DNA polymerase enzyme. The other component is primer. This primer is a short DNA fragment which initiates the DNA extension, DNA synthesis reaction. So in the PCR, two primers, that is forward and reverse primers, they are used, and they will be binding to each of the single stranded DNA which are flanking to the target DNA sequence. And these primers generally they are complementary to the template DNA sequence, and they are designed by analyzing the template DNA sequence carefully, and sometimes they are also designed using the freely available softwares. So certain criteria are taken into consideration while designing a primer. The first one is size. The length of the DNA fragment should be between 18 to 24 bases. And the second one is primer melting temperature. The primers with melting temperature in the range of 52 to 58 degrees centigrade, it is used in order to have a better annealing efficiency, that is binding capacity to the target DNA sequence. The GC content. The GC content of the primer should not exceed 40% because it promotes non-specific binding to the other regions of the target DNA sequence where there is G and C content, it is abundant. There this primer will be going and binding. So our primer should bind only to the target sequence. So in order to prevent this non-specific binding, the GC content of the primer should also be taken into consideration. And the next feature is primer secondary structure. So if there is a complementarity in the bases within the primer or between the primer molecules, it promotes this intramolecular base pairing or intermolecular base pairing. So in order to avoid that one, so you should take care that the complementary bases are not present within the primer molecules. Then the next one is primer template homology. So no homology within the template other than the target site should be there in order to prevent the non-specific binding. All the components which we have discussed to make the reaction mixture, and this reaction mixture is taken in PCR tubes, and the PCR tubes are inserted in a machine called as a thermal cycler. So this is the machine which carry out the PCR reaction, and this works on the principle of Peltier effect. So here you will be setting a program where the temperature is increased and lowered in a particular way. So these PCR tubes are kept in thermal block with holes. So this is the thermal block with holes where this PCR tubes are inserted, and the three events are that is the denaturation, annealing and extension. They are repeated for 30 to 40 cycles to obtain the DNA in million number of copies at the end of the reaction. So these are the steps in a typical PCR program. So initial denaturation at 94 degrees centigrade for 5 minutes, then final denaturation at 94 degrees centigrade for 30 seconds, annealing at 55 degrees centigrade for 30 seconds, and extension at 72 degrees centigrade for 1 minute, and final elongation at 72 degrees centigrade for 5 minutes. And at the last after 30 cycles of this three-step reaction, you will be getting millions of copies of DNA which are stored at a temperature of 4 degrees centigrade. And now let's see the methodology and discuss about each step in detail. So the start of the reaction, you will be having a single double-stranded DNA molecule. So when you heat that mixture at 94 to 96 degrees centigrade for 10 minutes, you will be seeing the complete denaturation, that is the double stranded is converted to single stranded DNA. So in the denaturation step, this is the first step where the double stranded DNA is converted to single stranded DNA. In the second step, it is called as annealing or binding of the primer. So when you decrease the temperature from 50 to 65 degrees centigrade, the primers, that is the forward and the reverse primers, they bind to the 5' ends of the target DNA, and then it initiates the chain extension. So that is the third step. The tag DNA polymerase, it helps in the synthesis of the new strand from 5' to 3' direction using the primer and the building blocks, that is the DNTPs. So on an average, DNA polymerase adds about 1000 base pairs per minute. So in this manner, the chain extension will be occurring, and these steps 1, 2, 3, they makes one cycle, and in general 35 to 40 such cycles they are performed in a typical PCR reaction. And after these cycles, let's say that the 40 cycles they are completed, the reaction is held at 70 to 74 degrees centigrade for several minutes to allow the final extension. That is, if ever there is the extension of the chain is not completed, if you incubate the mixture at 70 to 74 degrees centigrade, all the remaining DNA will be fully extended, and at the end the reaction is complete, and the resulting this amplicons or the amplified nucleic acids, they are stored at a very low temperature, that is at 4 degrees centigrade, until further experiments are carried out. And these are the entire steps which we have discussed. They are represented in this picture, and you can view these steps in the coming animation.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Let's see about the application of PCR. So the PCR is applied in various fields of life, like in food science, forensic science, disease diagnostic, and plant and animal tissue culture, and also in the identification of new species. And let's see the applications in detail. So it is used for the diagnosis of genetic disorders like cystic fibrosis, muscular dystrophy. So it helps in the determination of whether the individual is homozygous or heterozygous for a particular mutation, and this information, it is useful for genetic counseling of the parents. It is also used in forensic studies, in particular in the DNA fingerprinting. When the sample is very low, then you can amplify the sample obtained from hair, blood or bone or semen samples using this PCR technique, and it is also used in designing this VNTR markers, variable number tandem repeat markers, which helps in easy detection of the individuals in case of the illegal paternity cases or in criminal cases. Then this particular PCR, it is used as a diagnostic tool. It is for detecting the bacterial and virus infections. It plays a prominent role. Besides detecting the presence of the pathogen, it also quantifies the amount of pathogens which are present in the patient blood, and also it helps in monitoring the progression of the infection and how the patient is responding to a particular drug. So if you see this picture, the sample is taken from the patient, and by using this PCR technique, you will be able to detect whether that particular infectious agent is present in the sample or not. With the presence of the band, it indicates the positive test, and whereas the absence, it indicates the negative test. And the size of the band, it determines the amount of the pathogen present in the sample, and also the progression of the disease can be detected based on the size of this DNA band. And the other applications in gene cloning experiments, it is used to amplify the desired gene, and that is for the amplification or multiplication of the desired gene from the source. This techniques plays a very key role, and also it is used for the identification of the transgenic organisms. So this PCR, it acts as a selectable marker, and it is used to select the transformed cells. Whether that foreign gene is inserted or not, you can easily detect by using this PCR technique. The next application, it includes the gene expression studies. That is the PCR in the form of the reverse transcriptase PCR or real-time PCR. You can this one can be used as a scoreable marker, which is used to study the level of expression of the gene. That is, you can quantify the gene expression using this RT-PCR technique, and also the different levels of gene expression can also be studied using this RT-PCR technique. Next application is phylogeny studies. The phylogeny of organisms like the plants, animals, and other lower organisms, it can be traced out using this DNA analysis, and the origin of unknown samples, then that is, it can also be detected using this PCR technique. Like this, there are different applications, and in the last slide, let's recap some of the main points. That is, the PCR, it mainly involves 3 steps, that is denaturation, annealing, and elongation. Denaturation, it includes the conversion of double-stranded DNA to single-stranded DNA at the temperature of 94 degrees centigrade, and annealing means, that is here you are seeing the temperature at 55 to 60 degrees centigrade. The primers, they get annealed at the 5' ends of the target DNA. And the next step is the C. This one is the elongation. That is, once the primer, it goes and binds to the template DNA, then the DNA chain extension will be occurring in the presence of DNTPs and the tag DNA polymerase. It will be occurring at the temperature of 72 degrees centigrade. In this manner, these 3 events, they make one cycle, and like this 30 to 40 cycles are run in a PCR reaction to get multiple copies of DNA. So thus PCR is used to amplify the DNA into a large number of copies. Okay, with this I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.